Okay, I think we can get started now. So welcome everyone, welcome back. Today is our 5th day of our session, and hope you guys have enjoyed the session as much as I have. And I've learned a lot these past 4 days, and I hope you have too. We're gonna have a BIBEK give us a lecture about Introduction to R. So just a few instructions, your mic is muted because we have a lot of participants, and you can only type in your questions. All the sessions are recorded, and it's going to be sent to your email. You should have already received them. It's to the email address that you have used for registration. But BIBEK is going to be answering your questions live today. So please send the questions in during the session. So I was just thinking about the history of bioinformatics. So I was going back to the date when I started learning bioinformatics, and my dad actually introduced me to it. He brought a paper that 15 years back, and he said there's a new field called bioinformatics and it looks really promising, and I think it combines all the subjects that you really like, and you need to check it out. And there's no looking back. It's been amazing. And I wanted to read about the journey, and I read this really cool paper, A Data-Supported History of Bioinformatic Tools. It kind of shows you how it's evolved from back in 2000s and how it's come right now, how many tools have been developed, how many databases have been developed. So I was just looking into that paper, and I thought it's a really interesting thing to show with the timeline. As you can see, I guess the field started back in 1990 with just a very small amount of tools. And you can see as years have gone by, how many tools, how many databases has been developed. And it's a huge field where we can get lost and we don't know where we are, and there's so many databases and tools out there. We're not sure what we're supposed to use for analyzing our data. How do we get started? Like sometimes we just get lost in the amount of information. So I thought this is a really cool paper to read. And also I wanted to show you about this page that they use that shows all the different, I've sent you this link. So it kind of shows all the databases and tools. So let me just show you. So it shows the subsets of bioinformatics, how there's genomics, how there's proteomics and epigenomics. So there's all different things. So if you click on them, for example, I choose genomics, right? So you go in and you can see all the stuff that says, what are the tools used in genomics? What are the sequence? And what are the structures, editing? So it kind of, I felt like this kind of gave a little bit idea where we don't get lost as to what's out there, where do we get started? How do we start the analysis? So I thought this is a thing for you guys to like, maybe if you want a metabolomics. So what is metabolomics? What are the different tools that are available? So definitely check this out and see. And this paper also discussed about the evolution of some of the popular programming languages. As you can see from 2000 to 2015, you can see most used is R and Python. So that's why for bioinformatics, we prefer those two programming languages, because we're able to discuss a lot of analysis and stuff with R and Python. And this data shows the publications that's been done from 2000 to 2016. So the publications, as you can see back in 2000, they only used almost like one tool or maximum of 5 tools that was used. But you can see as years go by, people to publish a paper, they have to end up using almost 11 to 20 different bioinformatic tools to come up with a significant conclusion and to publish a paper. So this provides us the challenge of where we get started, like to actually finish an analysis. What are the multiple tools? What sequence are we supposed to use? How do we get started from BLAST to what structural database we need to use, and what is the path that we need to take to come up with the final paper? So this is the challenge that we pose today, because back it was very easy because there were not a lot of tools and databases available. As the amount of database and tools keep increasing over the years, we also have this challenge of what we are supposed to use, what's out there. So from the previous sessions, I received a question, which was, what skills should a bioinformatician have? I would say some aspect of molecular biology, like know why a DNA sequence is transcribed into RNA, why it's translated into a protein, why this is important. also have an experience of at least 1 or 2 major molecular biology software packages, know how to do sequence analysis, know how to do molecular modeling is really helpful. Also be comfortable working in a Linux or Unix environment, and have some programming experience, R, BioPython, C, C++, and Java, and having some basic knowledge of statistics. I would say these are the minimum to have these are the minimum skills to have as a bioinformatician to be successful and to go in the industry. I would say this is the minimum qualifications that you need, and then you can go and develop yourself and read more and learn more skills. Another question from the previous session was, what are the real industry requirements? I would say for a computer programmer who is trained in statistics, they actually end up developing these software tools and applications that you use on a day-to-day basis. So they develop these tools and softwares from scratch, and this has been used by the biotechnology companies. And these software programmers, they start cross-training in life sciences. So they learn a little bit of life science, so they understand the data better. And people with background in life sciences, they end up using the program software packages and databases that's been developed by the computer programmers. So they basically end up using those tools and softwares to translate the information so you can make new molecules and drugs. So these are the 2 broad fields, I would say, and this is what the industry is looking for. A real computer programmer who can develop these tools and applications, has knowledge in knowledge in molecular biology, and people with life sciences background who can use these tools and programs with confidence. So this is the 5th day, so I want to thank a couple of people who made this happen. I want to thank everyone who's registered and participated. I wasn't expecting this. We've had almost 800 or more registrants and participants, and I've been really grateful for that. I want to thank my university on taking my idea and actually making it a reality. I want to thank Therese, my associate dean, Alex, Jennifer, Mark, Carl, Monica, Kathy, and others. I'm sorry if I've left any of you. You've made from advertising to making this happen. I'm not familiar with Zoom, so making everything work. So you guys have been amazing. So thank you so much. And next, I want to thank my instructors. They've taken their time. They've come up with these lectures, and I really, really want to thank them. And I want to thank my family, my husband, my parents who've supported me, and especially my in-laws, because they're here taking care of my kids so I could do this. Thank you so much, everyone. My mom always says this when I think that I know a lot. She tells me, known as a drop and unknown as an ocean. And it's true in our case, especially in bioinformatics. We only know a drop. And no matter what age you are, no matter where you are, keep learning, keep pushing hard, and have the thirst for knowledge. Keep learning every day. And now, with that being said, I want to introduce BIBEK. I want to say that he has a PhD in biomedical data scientist. He's working as a postdoc in Stanford University. His research focuses on developing machine learning and statistical modeling for personalized medicine. He's worked extensively in research projects for text mining, and he's going to teach Introduction to R Programming with us in the fall 2020. We're very excited to have him. He is a typical example, I would say, because his background has been with a lot of computer science, and he's totally doing a lot of bio data now. So Vivek, do you want to take over?
Yes, thank you, Zanani, for a great introduction to the field of bioinformatics and your experience working with this as well. So let me start by sharing my video first, and then hello everyone. My name is BIBEK, and as Zanani just mentioned, I am a postdoctoral researcher at Stanford University, working in biomedical, and yeah, very happy to be here to talk about introduction to this field, to this emerging and growing field, with the powerful technology we have, which is R. My slides will be available to you. I just shared it with Monica, and Jen, be able to get it from them. I'm just confirming nevertheless before going into the slides. Let me see it one more time. Yes, so biomedical data science is a big field in which different disciplines come together, biology and like the field of statistics, computer science, and also the domain knowledge expertise of fields like epidemiology and several other fields. I don't know if you can see my screen, if one of you can confirm.
Hello, BIBEK. Yes, we can see your screen. Thank
okay,
you.
yes, that would be great. Yeah, so in this course, we'll be learning about how to use R to explore the basic techniques in bioinformatics. It is such a vast field, we'll not be able to cover everything, but the fundamentals that we need to get started in bioinformatics and to make professional stunning visualizations, to get a first dive into the field of data science, especially biomedical data science. This course will provide you all the fundamentals that you'll be needing for that kind of a journey. So as I was just mentioning a while ago, the skills that you need to be a bioinformatician or a biomedical data scientist, it's the coming together of computer science, statistics, computational biology, data science, and these days even machine learning and AI, right? There is no limiting the field, because as science in general progresses, so does the scope of bioinformatics. And to look at how this field has been expanding with the advancement in science in the recent years, we can have a look at this is from CNBC in 2018, and this is even more true in recent times. With all the problems that we're facing, the opportunities also arise. In that, the biggest industries, the industries with biggest amount of growth, both in terms of revenue, employment opportunities, and potential for your growth, for your professional growth, are healthcare and technology, right? And then when you look at the specific areas in these two fields, so in terms of healthcare, you have biotechnology, health data management, and then I have a comment from someone. Can you put my slides on presentation mode? Oh, I think it was for Zanani a while ago. I think my slides are already in presentation mode.
Hello, BIBEK. Yeah, your slides are in presentation mode, so you're set to go.
Okay.
Yep.
So if you look at the specific fields in healthcare industry, the areas that are seeing the biggest growth are data management, personalized health, biotechnology, the very fields which are directly intersecting with bioinformatics. And when you look at technology, it is all these emerging technologies as well. For example, cloud computing, machine learning, big data, and so on. So bioinformatics is really at this very, very emerging and growing intersection of the two most biggest thriving industries of our times. And if you look at job market, especially in the US, the places where you see the biggest growth, biggest opportunities for bioinformatics career is in fact California. If you look at this map, there are some states in the East Coast, some in the South, but by far the darkest color here in terms of job market is California. So let's have a few more slides to look at, what are the most in-demand skills in the job market these days. So this is from the banking giant BBVA, the top paying tech skills and experiences. R comes to the second on this list, among the programming languages you have. So there are all the other languages like Python, Ruby, but R, because it is so specialized and it is so versatile in dealing with all the different data sets, it is the skill that you'd want to make yourself familiar with if you want to get into this field. And this is from the website Stack Overflow. which is a large community of programming question answers. You see that the popularity of R, which is the blue line here in this chart, has been growing at a very steady pace since 2012 until this chart was made in 2018. It is going up and up throughout all these years, and more so than any other language during this period. So to summarize, I think, and I think you'll agree with me when I say this, that we are at the right place and in the right time to be looking into biomedical data science, and specifically through the lens of R. You know, how do we do biomedical data science with R? And this is what this course is going to be. So to discuss a few more things about what R actually is and what, you know, it might be sometimes a very confusing terminology, because R does not mean one thing all the time. It is, for the most part, it is an environment for statistical computing. And this environment encompasses several different things. R is also a programming language in that you can write very specialized code, you know, computer code to do several tasks. So this is a programming language as well. And in addition, it is also a software for generating all these software, all these summaries and graphics. It comes with a GUI, a graphical user interface, much like any other GUI you may have on a computer, like Microsoft Word or Excel. So that tool is also R. And in addition, R is also a big community of experts, novices, and people with, you know, various experiences level that are online and helping each other in the spirit of open source community. This is unlike many other platforms there are for statistical computing, and we'll be looking at some of the differences in the next slides. But what makes R really great is this community that makes it, you know, like up to date all the time and available with all the breakthrough, you know, statistical tools. That's what makes R stand out from the rest. And if you look at this picture on the right hand side, it kind of shows you the summary of the different components on a typical R user interface on your computer. And you can see already here, the tool that you're using to do all this analysis itself is part of the R environment. And then on the top left here, you have the script, which uses the R programming language. It has a specific syntax to do different things. So the script itself is R. And then there is the console here, which shows all the outputs from your script. There is the graphical output here, you know, all the charts and graphs that you can make. And then there is the data elements that are also part of this ecosystem. So all of this thing together makes, you know, the R environment. Now, why R and how is it different from other things? You know, R is not the only thing you can use for doing statistical analysis and also for doing bioinformatics. For sure, you can use several commercial and also, you know, proprietary or open source tools. I'm trying to give a very high level summary here of some of the tools that we have. The most familiar to all of us is Microsoft Excel. And I'm not trying to say here that any of them is not good or not suitable for the task at hand. The choice of the tool depends on the task that you want to accomplish. And any of these tools might be perfectly fine for different tasks, but there are specific circumstances when you should choose one versus the other. And there are definitely arguments for choosing R in comparison to any of them. So for example, if you have the question, why should I use R and not Microsoft Excel? I think Excel is a very nice tool. It is very friendly to most users. You don't have to learn a new language or anything. It is very intuitive, but it comes with some drawbacks that it cannot handle a lot of large files. It cannot, yes, and there are some questions about my slides. I'll be coming to the Q&A just at the end of the next 2 or 3 slides, so bear with me until then. So R has the advantage in comparison to Excel in that it can handle really large files. And in the era of big data, and when you don't have small data sets anymore, you have genomic sequences from several different species, or even very large sequences from a single species. Microsoft Excel is not meant for such tasks. And you may even try, you may try to open a data set of a few, not even a few 100 megabytes, maybe even 50 or 60 megabytes, and Excel will already start feeling slow and unresponsive at that time. And another advantage of R is that you cannot use Microsoft Excel on really all the platforms. When I mean all the platforms, I'm not talking just about the desktop computers or laptops. There are even very small hardwares nowadays that can be used on sensors or on spacecrafts, or there are all these different kinds of Internet of Things devices where you just don't have the resources to run this GUI system. And R is again useful in these cases. And when it comes to the sophisticated state-of-the-art leading edge statistical analysis, R is where it shines. That's where R shines. So yeah, if you need to work with big files, latest statistical analysis techniques, and on different platforms, you should switch to R. But otherwise, if your data sets are small and if it is run-of-the-mill, simpler analysis, Excel is perfectly fine. Similarly, there are other tools like Stata, SPSS, SAS, and so on. The biggest advantage in comparison to Stata, SAS, and SPSS is that they usually cost a lot of money for licensing and for use. R, in comparison, is not only free to use, you don't have to pay money to use it, but it's also free to modify. You can also add your own code to R because it's an open source software platform. And that's why most of the scientists and academic researchers who do statistical analysis, they write their code in R and make it available for everyone. That's how you have the latest analysis algorithms and tools in R, which you don't have in other platforms. Yeah, there's a summary of some advantages of R in comparison to the different popular platforms. Definitely, this is not an exhaustive list. There are also some downsides to R, which we can also talk. Let me take some questions at this moment. Let me pause for a while and take some questions. If you have some questions, please feel free to send them. We'll be doing this for the next 5 minutes. I can already see some questions. So there's a question from about the minimum hardware requirements to run all these kinds of data analysis. So when you say minimum requirements, there is not really a minimum requirement for running R in itself. And I'll show you in a few slides. R is able to run in really primitive and small and resource poor hardware settings. But if you have very large data sets, for example, right? If you are using gigabytes of data to do some statistical analysis, then you'll start to, grow the need for more expensive, more, powerful hardware. But otherwise, to just try on some small data sets, if you have a desktop computer that runs, the other softwares like Word or Excel, it is perfectly fine. That hardware is already enough. And then there is a second question about what are the advantages of R over Python? Okay, I think I'm in the perfect spot here to answer this question. You can see on the slides. The advantage with R is that it is more statistically oriented. Python is a general purpose programming language, and it has a lot of bells and whistles because it is meant to do so many other things, not only statistics. So you have to sometimes also learn parts of these other things to understand Python really well. And the downside of Python is that since it is not statistically oriented, you'll not get some of the very latest statistical libraries there. And when you get them, they may not have all the features. So our advantage in comparison to Python is really the specialization in statistics and its narrow focus on data analysis. But if you want to do other things together with data analysis, you can also integrate R and Python. It's not, it's definitely not the case that if you use R, you cannot integrate this with Python, because they're both open source platforms. They use common standards. They can read text files in a similar way. So you can switch over them, pretty effortlessly if you know both of them. So there's one more question about, would you consider the fact that R is vector-based to be a strength or weakness? I think when you're a beginner, it can be a weakness because you have to change your way of thinking. In that R organizes everything as vectors. But when it comes to doing statistical things, it is a really smart way of organizing data, because in reality, all the mathematical structures underneath deal with vectors, right? When you have large collection of numbers, they're usually treated as vectors and then as matrices and so on. So in the underlying, under the hood, it is a strength. But when we begin to learn, in the beginning stages, it may come as a hindrance. But again, the goal of this course is to get past these hindrances, to be familiar with these intricacies of R, so we can move on to the next stages. Yeah, all the questions were really nice. Thank you for that. And please keep sending questions. I'll be, stopping in between to answer them in a while again. So in comparison to all the other tools, what is the advantage of R? I've already gone over some of these points while answering your questions. It is the tool that is preferred by specialists. So if you are going beyond the basic analysis and trying to dig deeper into a field, many of the other tools will have limitations, and what you want will not be achieved by those tools. So since R is a tool preferred by specialists, if you want to do specialist tasks, you'll have a big advantage if you know R. And it is a very actively developed software, meaning that every day and every hour even, there's new code, new analysis tools, new visualization techniques that is being integrated to R. So you are getting all this, bleeding edge technique for free. And if you need help, there's a very big online community for support. I mean, for other paid software platforms, you usually pay a consultant who will help you with this. But for R, you can of course also have paid consultants, but you can also get this for free on online platforms. And the volunteers who are writing code in R will most often be very receptive to questions and will be answering them. That's a big plus. And also another advantage is that anybody can contribute and add more tools or more code to the code base of R. So if you have reached a point where you have a very specific improvement that you can introduce, you can be the author of that specific part of R. So you can join this online community in that way. And some of the advantages that we already talked about is that it can handle multiple and very large data sets. It can work with resources on the internet, meaning you can directly download a file from a web page and start working with it, right? And it has the latest statistical models. It is very easy to automate, right? You don't have to click buttons all the time. You can write code, and on the background, you can make it do things automatically. For example, every day you want to get the temperature and then do some predictions. You can write codes like that for R, which, for example, for tools like Excel, you'd not be able to do. One downside to R is that because of all these specialist and very niche application areas, compared to other tools, it has a slightly high barrier to entry, meaning it takes a bit of a time to learn and be familiar with doing things in R. But once you've crossed this barrier, then I think I've not met anyone who has regretted learning R, although in the beginning they might have had some difficulties. So when you talk of specialization in R, you can look at this graphics again from Stack Overflow by, so it breaks down the industry of people who visit questions related to R. And you can see that the large majority of people are from academic institutions or healthcare, which means they are researchers or they are professionals working in bioinformatics, and then government, consulting, and so on. So it is really popular among those, who need to do specialist analysis, statistical analysis. So yeah, I think I've stressed enough about why R can be helpful for you, how it fits in the general landscape of bioinformatics. Let's get a feel of how it works. So the most basic way you can interact with R is through a command line or a terminal. Maybe some of you have already used command line tools in the past. If you have not, it's not necessary that you must use command line to interact with R. But if you don't have, as I was answering a question before, if you have a very limited hardware, a very small amount of RAM, memory on your computer, you can run this command line as a command line tool because it really does not need a lot of resource. And to give you an example, let's have a look at this computer. This is Raspberry Pi. It's a credit card size computer. As you can see, it is smaller than your palm. So, and this computer costs less than $50 to set up and get running, right? So it does not need a monitor to run. You can just plug it to power, as you can see here. On this tabletop, you have the Raspberry Pi connected to the internet and running, right? And when you have tools like this, you need really resource efficient and lean tools, and R can be used in such tools here. So that is the most basic, most bare bones format of R which you can run. If you know R, of course you can do this, but in the beginning you don't have to. We have different interfaces and tools to make life easier when we are beginning. You can use a normal programming language editor to write code. If you've written code in Python, C, Java, or even HTML, it is a similar platform. You can write code like this, or if you prefer a graphical user interface, there's a very popular interface called RStudio, where you can write code and immediately look at what's happening, what kind of data it is creating, what's the output. It's much more interactive. So, and in this course, we'll be using some of these graphical tools that makes learning much easier. And once we learn this, we can always go back to command line because it's not much different. There's a question from the audience. The question is, can we work with R in any other programming environment? So I don't know if I understand the question properly, but R is itself a programming environment. So if you want to use R in any other programming environment, I'm assuming, for example, you want to call R scripts, R code from some other programming languages. Yes, you can do that, but I don't see an advantage of doing that unless you want to integrate something you've done in R into some other programming languages. But there may be limitations. You may not be able to integrate this with Stata or SAS, for example, right? So you cannot do this universally, but for many languages, yes, you can integrate R with other tools. Now, so R composes of two main components. the one that I just showed you in the previous slides, the tools that you use on your computer, they are only one part, right? And in its most barest form, R comes with just the most basic functionalities. You can read a file, you can do basic statistical summarization and so on. But there is so much of other kinds of needs that you want to address. For example, you want to do genomic sequence analysis. It is a very specific tool and not everybody may need this. So R does not come with tools for genomic analysis by default, okay? You have to use small tools, and they are called libraries in the R lingo. There are several different libraries. There are thousands of libraries, and each library is made for a specific task, and they add all the different high-level functionalities that are needed in your day-to-day life. So here are some of the most used libraries on the figure here. Some of them are used for graphing, plotting charts. Some of them are used for doing machine learning. Some of them are used for doing predictive tasks. Some of them are for bioinformatics analysis and so on. And all of them are also available for free. So you can install the ones that you need. You can use them. If you need something else, you can install them. If you feel that there's something that has not been solved, you can write them yourself. That is the nice thing. So what about bioinformatics, right? There are several tools, several libraries also. Some of the example cases that you can do in R using all these libraries is that you can do all the statistical summarization that you need to do from big spreadsheets. You can create visualizations. You can do like genomic analysis, proteomics, all the molecular data analysis. You can also use R for clinical studies. And moreover, you can do social media and text analysis, right, which is also sometimes part of bioinformatics. If you want to see, what kind of disease trends are happening on social media, you can also do that with R. You can use R to look at electronic health records, epidemiology, and population health. There's a question about the best library to run and visualize PCA. We'll be discussing these things in the course because there are so many. There's not only one library that you can use. There are multiple, right? And we'll be discussing them. We'll be discussing the libraries and tools that we need during the course. So I'd hesitate to say one library is the best because it again depends on the task that you want to do. But definitely there is PCA, there's also all sorts of other dimension reduction techniques that you want to do. Here's an example of one package. Okay, this package is called KUENM. There's a question about when the class will start. I think Zanoni will be informing you, or you can talk about this at the end of this presentation when we'll have Q&A about the logistics of the course. So let's look at the KUENM package here. It's a package for doing ecological niche modeling. And what it does is this one package gives you the ability to prepare the data, to clean the data, to organize it, to build a model and to calibrate, and to do your final decision analysis. So for example, the summaries this package creates, it can be used for scientific presentations or for your professional work, right? It has statistical validity. You can look at the separation of data and so on using the standard, standards of the industry. And you can also do some cool visualizations. In this case, the library integrates your data with the global map or even the map of a country here. In case of this right-hand side, the map of Cuba, and on the left-hand side, map of the world. It is visualizing the occurrence of two different species, tick and toad, over these maps, right? You can do all this using using R. And what will this course cover, right? So this course will not be going through all of this in one course because it's not possible. We'll be needing to do small steps. So maybe in multiple courses, 2 or 3 courses, you can accomplish all of this. But in the beginning, today, I'm going to present you case studies, 2 use cases using the COVID virus, right? How can we do, what kind of things we can do, to give you some examples. And at the end of this introductory course, you'll be familiar with all the tools necessary to do this kind of analysis. So let's have a look at the analysis that we'll be doing in this course. I'll take Q&A in maybe two more minutes, so keep your questions coming. The examples that I'll be presenting today is, the explanation that I'll give today, you may not be able to understand all of it. Don't worry, because that's not the goal. The goal is to see what R is capable of doing, and after a few stages of learning, what you can do. Because you don't have to, it's not mandatory that you understand the inner workings of all of these techniques, right? Sometimes it helps if you do, but even without a very deep understanding of the subject, you can work with the data and have fun and understand what's going on. So in this presentation, I'm going to talk about a very brief summary of how to do genomic data analysis. And a second example is how to analyze the spread of disease. And you can see examples of this analysis in popular media these days. There's a website called Next Strain. If you go there, you can see all the different strains of the coronavirus since it was detected a couple of months ago. And using the genomic sequences of the viruses, what scientists are trying to do is they're trying to trace the origin of virus. Okay, we'll look at an example. And this is a very helpful tool. It's also helpful for analyzing the ancestry or hereditary information amongst people, also to look at which kind of diseases are caused by what kind of genes, right? And then the second one is a slightly different analysis. We'll be looking at how the disease has been spreading in different countries. So at this point, I'll pause to take 2 questions. One is, how resource intensive is R compared to Python? That's so there is no one universal answer, because as I said, in both its barest form, when you don't install anything, both R and Python can run on pretty much any standard hardware, any standard, even the smallest computer can run, even the Raspberry Pi can run both Python and R. The resource intensive part comes when you do the actual data analysis. If your data set is very big and you want to do complicated analysis, then you'd be needing a sophisticated hardware. But otherwise, I'd say both of them have pretty similar requirements when you use them in their most basic forms. Then what you do on top of that is what's going to determine the resources that you'll be needing. So I won't say R is more resource hungry or Python is more resource hungry, but if you do some, like heavy computing, that's where you're going to need all these additional resources. There's another question, which is I think it's going to be useful to many of you. Is there any online environment tools where you can use R? Yes, you can definitely try R on Kaggle. If you have heard of this website, I think the website address is kaggle.com. You can even try different competitions there. There's another website called Google Colab, where you can also try R without installing it on a computer. Which has a faster runtime between R or Python? Now the answer depends also on what kind of, so R really signs in vector-based operations. So for data structures that are organized as vectors, which is most statistical data sets, R is faster. For several other things, if you want to do deep learning, for example, then Python will be faster because it has all the tools needed to run on very specialized hardware. Again, I think it's not fair to make comparisons like this, which is faster or which is more efficient. It's all depends on the task that you want to do. And sometimes even if it is slower, it might be better to use the slower tool because if it does your, if it solves your task. Now, let me go back to the final part of this presentation, the basics of R. So in R, everything is organized as a vector, right? So you can see on this top left, a vector, if you can recall from your preliminary math lecture, is a collection of numbers, collection of different data in the sense. It does not have to be always numbers. It can also be letters, like alphabets. So even text is organized into vectors in R. If you read a file, the characters of the file will be loaded as a vector. So yeah, so everything is a vector is really what you need to know. That's the first thing you need to know when you learn R. And then these vectors are organized into matrices by combining different vectors into rows and columns. And you can organize even more matrices to make an array, right? This is a 2-dimensional array, this is a 3-dimensional array, and so on. And what you call spreadsheets in Excel are data frames in R, or a table, right? Here you have rows of data organized into different columns. And then from vectors come all the other sorts of things like lists. Lists can be of same data types or different. There are dictionaries and so on. So we'll be learning, our journey into R will start from these basics primitives. So we call them primitives. Vector is a R primitive, and using vector, you can build all the other things. So let's have a look at the examples that I wanted to show you. So why is next strain used? You can have a look at the tweet from today, and you can see here all these colors denote the similarity of the coronavirus gene sequence, right? And by looking at how similar each gene is to another, like gene sequence, you can trace the origin of this virus. So the virus that came to the US most likely came from Europe because it is most similar to the sequence of virus found in Europe, right? And we'll be doing this analysis right in a moment. And it is really useful to look at genetic epidemiology of different pathogens. And the second analysis is looking at, how fast or how slow a disease is spreading in different countries. So I'll switch over to my R environment so you can have a look at some of the analysis that we can do. I'll stop sharing my slides and move over to my R environment. Give me a moment. So, yeah. Okay. Can you see my, yes, can you see my R screen?
Yes, we can.
Yes, okay, cool. So I've already loaded, I've already written the code here, but we won't have time to go through each of the code blocks, but I'll tell you briefly what everything here is doing. So this whole window is R Studio. It's an environment for doing R programming, and here there are some segments here. This part here where my mouse is moving is where you can write your code, and you can see the output here below in this section. And you can see the plots, the files and so on. Here, you can use mouse click and, you can use like a GUI to install libraries, right? These are all the libraries that I have to look at your plots, to look at the files on your computer, to look at even help, right? If you want, someone was asking about PCA, right? You can look at how to do PCA here. And so it is a combination of a graphical tool with a programmatic environment. So let's look at some components that we talked about. We talked about libraries. So I'm loading some libraries to R using these commands here, and it's a very simple command. You just use library, then you give the name of the library, and then so these libraries are loaded onto the system. And yeah, you can see all of these libraries are loaded. This is the output that you see. I'll move this. Yeah, I'm taking whole screen here. And then I have already downloaded the coronavirus gene sequences on my computer from the NextStrain website. I'm going to read this file and load it into a variable called ncov sequences. And once I do this, it has different. So here is the data set that I loaded, and here is what it looks like. If you click here, you can even see. So there are different sequences here. This gene, the virus gene from Australia, Japan, Shenzhen, all these things. And here is the actual value of this genome, 80, all these nucleotides in this genome. So it is a vector, it is a character vector, and it has these many entries, right? As we discussed before, everything is a vector. So you can look at all the data you're creating as you go, right? It is as friendly as that. And I'm only taking, because, for the sake of simplicity, I'm not looking at all the genomes here. There are too many of them. I'm selecting 15 of them, okay? I'm only looking at 15 of them so we can visualize them easily. I'm giving them some names, right? So these are the 15 genes that we'll be looking at. Some are from China, some are from Korea, Spain, US, Taiwan, and so on. And then I'm going to create what is called a k-mers of genomic sequences. I see. Yeah, so when I created this, I forgot to move over some other files. Let me fix this in a moment. Okay, and then here you have, so what I do is I create the, from the sequences, I create sequences of 5 nucleotides. And then from these 5 nucleotides, I'm calculating the similarity between each of these sequences, right? When we create similarity in a matrix, I compute the similarity in a matrix. The more k-mers overlap between 2 sequences, the higher similarity you have. And then what I do is I'm using a function from a library, right? I don't have to write much sophisticated code to create this phylogenetic tree here. I'm calling a library function with this distribution I just created with the distance matrix. And then you can see here, it is giving me the phylogenetic tree of this 15 genomic sequences. And by looking at this visualization, it is very similar to the one you saw on NextStrain, in that the sequences from Spain are close to each other, and the Spanish sequence most likely came from Taiwan, because it is very similar to the viral genome from Taiwan, found in Taiwan, which is similar to the one found in United States, California, right? So you can look at all this phylogenetic tree to do some really interesting detective work of where a disease, where a pathogen spread from. So at this point, let's also look at some questions here. So there's a question about what kind of input file is it? This file is the genomic sequence file published by Next Strain. You can also download it on your computer. Go to their website, which I showed in the slides. It is Next Strain from University of Washington. They publish all these sequences. So it is this FASTA file. There's another question about if the file is aligned or not aligned. Aligned, you don't need, so in the algorithm that I'm running here, you don't need aligned sequences. Unaligned are also fine. And oh, they have aligned it, but in this file they have not. Okay, I mean, and then there is another question about what is the current scenario between R and Python integration that will be changing the environment. Yeah, R and Python, you can run R code from inside Python, but you'll not be gaining all the advantages of using R as a separate tool. Okay, but you can definitely do integration. You'll be limited in what you can do, but definitely there's integration. And then yeah, so I'm for the interest of time, we're very close to the end of the this lecture. I'm not be talking in depth about the kind of data we have, and also not about the biology of this. The goal is just to show what you can do with really some basic R skills, and what's the first steps towards doing that. Can you run Python chunks in RStudio? I've never done that, but I think there must be a library to do that, because for everything there's a library. So I believe there is, but I've never done that. So yeah, so I'll not be, let's not go over the biological details of the biology here. Because I want to move to another example. This example is about, so here, this is the output we want to produce. We want to produce how many deaths there are in different countries. And when you think of this, this looks like a really complicated analysis, but the amount of code that I wrote to do this is only this much. Okay, I'll be explaining how I did this. There's a library called PH.D COVID-19 that provides the data set for deaths from different countries. And once I install this library, I can only load the deaths from these countries: Australia, Belgium, Brazil, and so on. And I can ask this library to plot it for me. You can, of course, do this in a different way by writing your own code. But usually, if people have written some nice code to do something really handy, it's a good idea to reuse their code. So there's a question about KMR analysis, changing the parameters. I can change the parameters, but since it has to calculate all the sequences, let's not do that right now. Will you post the R code examples? It maybe if there's a possibilities, definitely. It depends on the logistics for the course. There's another question about, are there other GUIs for R you'd recommend? I think RStudio is a very good one. Otherwise, you can use Kaggle or Google Colab, which is online, which is an online tool that you can use. And there's a final question of how recent is PH.D COVID-19 data? I don't know that, but they have a nice website which you can go and have a look. If you just look for PH.D COVID-19, you can have a look. So I think it pulls data from JHU, Johns Hopkins University website. So it must be pretty recent. It does not build its own data set, but just gives you the option to pull data from JHU website. So, and then you can, there's also a functionality of exporting all this file as an HTML, as a web page or PDF that you can put up on your website or distribute to others and so on. So it is really versatile in what kind of analysis you can do using R. So yeah, I hope it was helpful. And please let me know if you have some questions, and if you have some specific needs or topics that you'd like to be covered in the course, definitely let me know. I cannot promise that we can do all of that, but I'll try to make it as useful to as big group of people as possible. Thank you. And please stay for Q&A. Zanani and Monica would be here, I guess, to take some more of your questions.
Yeah, I think we can take a few more if you guys have any more questions. Otherwise, let's wrap it up because it's already 6:30. So thanks, BIBEK. It was awesome. And I just had a quick question for you. If we say we publish a paper or use this R code from someone else, I know it's open source. Do we have to put them as a reference, or do we need to quote it? I wasn't sure,
Yeah, it depends. Usually you can just give it as a footnote, right? Mm-hmm. But sometimes some R libraries are published as papers also. So there's a code and there's an associated paper of how it is done. In that case, it is considered, and if your analysis depends heavily on them, it is considered polite to cite them. Yeah.
Okay. Thanks, BIBEK. We're really looking forward to your course in fall of 2020 is when BIBEK is going to start his course. So thank you so much for doing it.
Thank you. Yeah.
I just wanted to go over a couple of, things with you guys. So hold on just a minute. So yeah, make sure you guys fill the feedback form because we will love to hear your feedback on all the 5-day sessions. And I am always looking to hire new instructors. If you guys have a good background in bioinformatics and if you want to come up with a course for our certificate, please feel free to email me a resume and what kind of course do you think will complement our certificate program. If you want to do an elective or if you want to suggest something, please definitely go ahead and do that. Other than that, make sure you do the feedback, and thank you so much for joining us. I really enjoyed this past 5 days. This has been a new experience for me and I've really enjoyed it, and I hope you guys did as well. I want you to know that during this lockdown, you guys had a lot of choices, but you choose to log in and come with us and learn with us for this past 5 days, and we're really thankful for that. I think if Monica, do you have anything else or should we wrap it up?
Hi, Jenny. No, I think that's it. I'm sending a link right now for the lecture slides, the recordings. We should be able to update tonight's lectures no later than Monday, Tuesday of next week. Just check on the links that I just sent to the panel. Lastly, I'm going to go ahead and send the feedback form right now here. We'd love to hear your feedback so that we can continue to have these sessions in the future. And then we will send this information next week. If you don't receive our email, then you can request the information separately at extension@ucsc.edu. And again, I'll go ahead and submit that in the chat link.
Perfect. Thanks, Monica. You've been a great help for us. I just want to tell you guys that, hopefully when we come back, things will get better. And I'm hoping to have a session where we can discuss the amazing vaccines and drugs that we have designed using bioinformatics. So that'll be really nice. Till then, stay safe. Good night, guys. Thank you.